Hi everyone, welcome to Popping the Science Bubble. Before we start, I'm going to read our land acknowledgement. Berkeley Public Library buildings are built on the territory of the Huichin within the homeland of the Chochenyo-speaking Ohlone people. We acknowledge and honor the Ohlone people who have lived in Berkeley and throughout the East Bay since time immemorial. The library recognizes that Berkeley residents have and continue to benefit from the use and occupation of unceded stolen land, and we pledge to recognize the permanent and rich contributions that the Ohlone people continue to bring to the city of Berkeley and throughout the East Bay. We celebrate the strength of the Ohlone people and their culture. The Berkeley Public Library is committed to helping create a more just and anti-racist society and to furthering the visibility and celebration of our Ohlone authors who are specific to the East Bay and our Ohlone culture in our collections and programming. This is a living document that was co-written by Ohlone cultural leaders Vincent Medina and Louis Trevino and the Berkeley Public Library. Welcome to Popping the Science Bubble. This is a monthly seminar series that aims to share new research findings from grad students and postdocs at UC Berkeley with the general public and create constructive discussion about a variety of science topics. We have 2 speakers at each seminar who will talk about their current research or a topic they find really interesting. The organizers are 4 graduate students at UC Berkeley: Kaylin Zwang, David Bretthauer, Sydney Hemingway, and Annika Velez. This program happens every 3rd Tuesday of the month at 5:30 at the library and on Zoom. If you're interested in checking out their past seminars, you can visit their website and sign up for the listserv to keep in the loop for upcoming seminars. And check out their YouTube page to view this lecture and see an archive of past lectures. So now that that's said, I'll introduce Sydney, one of our organizers.
Thanks, Molly. So to start us off tonight, tonight's first speaker is Kylee Hillman. Kylee was born and raised in Tallahassee, Florida, and attended Florida State University, where she majored in biology. After graduating, she got a job as a lab tech and solidified her love of and desire to pursue it as a career. Currently, she is a second-year PhD student in the Plant and Microbial Biology department at UC Berkeley. Outside of lab, Kylee enjoys painting, watching movies, reading, going to see live music, and doing anything outside. So with that said, I'll pass it over to Kylee. And just as a reminder, this is an open format talk, so we can encourage questions throughout the presentation or take them at the end. Go ahead, Kylee.
Let me see. Sure. Yeah.
We're just changing over for the speaker presentation.
Share the screen, please.
There you go.
You're seeing your
Oh,
presenter,
can you,
if you can-
Is it only going to do the presenter view?
I think if you share the second screen, can I?
Yeah, go for it.
So if you share, I think this one, that could work. Let's see. Does it work?
Yes.
So, thank you. Hi, everybody. My name is Kylee, and I'm a second-year graduate student at UC Berkeley in the Plant and Microbial Biology department. And today I'll be talking to you a little bit about my project called Sneaking into Cells: How Plants Are Helping Rewrite Their Own DNA. So to set the stage for this project a little bit, Heat and drought both cause, and other environmental factors greatly impact crop yield. So from this graphic, you can see how higher temperatures cause plants to demand more moisture, but when they're in a state of dryness, they can't really get that moisture. And that causes these plants to wilt and reduces our crop yield, which, as we have a growing society, it's really important to keep up with the crop demand. And so that's kind of what's inspiring my project, which is developing new tools to help engineer crops to be more resilient. And so to talk about this a little bit more in detail, I'm going to talk a little bit about some basic biology that kind of informs my project. And so many of you might remember all this, but if not, I'm just going to go over it briefly. So the central dogma of biology states that DNA, which is kind of the blueprint and everything that makes up our body, is transcribed into RNA, and then that is then translated into proteins. And you can kind of think about the DNA as maybe a recipe book or a recipe online. The RNA is as if you were writing down this recipe to take with you into the kitchen. And then your finished product, your cake, for example, is your protein. So naturally, in the process of DNA replication, mutations can occur. And these mutations, obviously, if they occur in the DNA, are then transcribed into the RNA and then translated into the protein. And sometimes this doesn't make any change at all. So you end up with the exact same protein as you had before. But other times, maybe you mutate it into some better function, like a 3-tier cake, or something totally different, like a pie. Humans have actually been taking advantage of these natural mutation for centuries in the form of selective breeding. So in this scenario, imagine you're a farmer and you have 3 plants. One here, which has your normal DNA with no mutations, and it has 2 large fruit on it. One maybe in the middle that has some sort of mutation you're unaware of, but it has many small fruit. And then our last one, which has another mutation and causes it to have very few fruit. So if you're the farmer in this situation, you might want to breed these 2 plants. And so in your next generation, you have more plants, ones that have slightly more and slightly larger fruit. And then you might want to mutate or breed these once again. And so you choose these 2, and then you end up with your final product that is many fruits, and they're quite large. And so this selective breeding has been really useful to humans, like I mentioned, for decades, for centuries, but it's pretty slow as a process. But with that being said, it also has greatly helped human society. And you might actually recognize a few things that have been selectively bred. So for example, this banana that you see on the right might look really familiar, but the one on the left is actually its ancestral how it is normally without the selective breeding. And so you can see there's this really thick green rind on the outside and really large seeds in the middle. And so throughout selective breeding, we're able to get a more enjoyable banana experience. And you can kind of see this happen again with watermelon. So this is actually a painting from the 17th or 18th century. And you could see this watermelon has this white, fleshy rind, and then these really dark black seeds. And then in our modern day watermelon, it's much more red with this edible part, and you can even get some without seeds at all. And then finally, we have corn, which many of you might recognize this version of corn, but it actually evolved from what we call teosinte, which is right here. And you could probably count the amount of kernels on that with one hand, versus our modern day corn have hundreds of kernels. And so I'm going to do a little activity here, and I basically just want everybody to answer this question. I'm just going to ask you to raise your hand when I call out the specific crop if you think it was not a result of selective breeding. Okay, so who thinks wheat was not a result of selective breeding? Okay, what about grapefruit? A little bit, yes. Sunflower? Okay, and then rice. So yes, it's a bit of a trick question because all of these are results of selective breeding. And like I mentioned, selective breeding has been really useful to us, but it's also very time consuming and not always super efficient. And so modern day molecular biology has come up with a new tool called CRISPR, which basically allows us to go in and make very specific edits to the DNA instead of just breeding things at random based on what they look like. So CRISPR is comprised of these 2 pieces. One is called the guide RNA, which kind of serves as the map. So it tells the protein where to go in the genome. And then the Cas is the cutting enzyme, and you can think of it as a pair of scissors. So when you put them together, you have the guide here that tells the Cas exactly where to cut in the genome. And this was actually pioneered by UC Berkeley's own Jennifer Doudna, who won the Nobel Prize with her colleagues a few years back for this work. And so CRISPR works kind of how I was mentioning before. When you put your guide with your Cas9, it kind of scans through the whole genome until it finds this specific match. And the matches are here in blue. So this blue region matches directly to this guide region in the RNA. And it kind of scans through, and once it finds it, the Cas enzyme actually starts to unwind this DNA, and it enables it to make a precise cut in the DNA like so. And because the cells really don't want broken DNA, they are really incentivized to fix it as quickly as possible. And when they fix it, oftentimes it can introduce mutations like we see here in green. So this is basically just a much quicker, more efficient way to be creating mutations like we were seeing from selective breeding. But there's one problem, and that is, how do we get this Cas9 machinery into plant cells if we actually want to use it for, making better crops? And so this problem arises because when we zoom in on this plant leaf here, we get our plant cell. And the plant cell has one really interesting characteristic called the cell wall. And this cell wall is made up of many polymers, sugars, and proteins that are very strong and kind of want to keep things out for the most part. So getting this Cas system in, past that cell wall into the cell to do its work is a little bit of a challenge. And so that's where the new tool that I've been researching comes in. It's called a cell-penetrating peptide, and it's comprised of small chains of amino acids. So amino acids are the building blocks of proteins, so you can kind of think of a cell-penetrating peptide as a small protein. They also have specific hydrophobic regions, as well as very highly positively charged regions. And so these kind of things don't really make much sense until you put them next to what a simple cell membrane looks like. So when we have our cell membrane, we have a very hydrophobic interior and a very negatively charged head group. And so as you can imagine, these negatively charged head groups kind of attract the positively charged residues in this peptide, and the hydrophobic residues in the peptide allow it to span across this hydrophobic interior, ultimately allowing certain cargoes, like our editing machinery, our Cas9, into the cell. And these cell-penetrating peptides have actually been discovered in plants. So most living things, or eukaryotes, have what we call developmental proteins, and they're responsible for how things look and how they develop. So for example, if you mutate one of these developmental proteins, say in a fly, you might get a fly with 2 sets of wings, or maybe in legs where they should have some antenna. So an example of this occurring in a plant is maybe the normal plant over here with 3 leaves. If you mutate their developmental protein, you might end up with a plant that has 5 leaves instead, because we're dysregulating this development. And these developmental proteins actually have a very specific motif region on the end of them that contains cell-penetrating peptides. So this specific motif allows it to be shuttled back and forth between cells as like a messenger, and we can actually use this as a way to get other things that we want into these cells. And so like I mentioned, plant developmental proteins have these specifically, and in this next slide, I'm gonna kind of take you through a figure from a paper that was from my lab and kind of show you how scientists prove that these actually work in plants. So on the left side here, we have what we call a gene fragment. And this gene fragment has a red fluorescent protein right here and a green fluorescent protein right here. And it also has these stop and starts, which are called promoters and terminators. And so the promoter you can think of as just the start. So that's telling the machinery to start transcribing the gene, the cellular machinery. And it also has, right before the green fluorescent protein, a stop. So in this specific model, the only thing that you'll see is the M-cherry, the red fluorescent protein. But then when you add another cutting protein to this, it actually cuts right here at this stop site. So between these 2 blue arrows, such that when you add the cutting protein, you will get both the green fluorescent and red fluorescent protein showing up in your samples. And so the way that scientists tested this is they actually looked at the plant leaf under a microscope, under a fluorescent microscope, to see if they could see the red fluorescence or the green fluorescence. And this first row here is mock treatment. So this is plants treated with only water. And you can see plants treated with only water do not have any green fluorescence. So this green fluorescence is this whole row here. And there's no green fluorescence here. You can also see down below that there is a little bit of the red fluorescence, which makes sense because without any alterations to our system, we should always see this red fluorescent protein. In the second column right here, we added just the cutting protein. So when you add just the cutting protein, you see still no green fluorescence and still only the red fluorescence in the red channel. And then finally, when we add our cutting protein to our cell-penetrating peptide, which we called CL-WOX, you can see that there is now green fluorescence in both channels, in the green channel and in the green plus red channel. So this indicates that we were actually able to successfully deliver the protein with the cell-penetrating peptide. And they also quantified this by counting the amount of positive nuclei, so green colored nuclei, which are these little dots you can see in here. These are the plant cell nuclei, and they counted however many of them were in each sample. And in both the mock and protein only, they got no fluorescence, but then in the sample with protein and peptide together, they were able to get an increase in the fluorescence. And so hopefully this kind of showed you all that we can potentially use these plant-derived cell-penetrating peptides as a way to get various protein, DNA, and other helpful molecules into plant cells and help them be more resilient in our changing climate. Yeah. Yes?
So does the model, so that's a natural process. And then your model replicate that or artificial?
So what's natural is the protein itself shuttles from cells to cells. And so what we kind of did is hijacked what was actually used doing the shuttling and attached it to a totally different protein to get it into the cell. And so it is utilizing some of those natural processes, but the cutting proteins that we're attaching to the peptides are not always situated like that, if that makes sense.
Okay,
Mhm.
hijack vehicle.
Yeah, kind of, exactly.
And then with the CRISPR
Mhm.
model or method, I'm having trouble understanding what it goes in and it changes what one strand of DNA or.
Yeah, it creates a double-stranded break actually. As it depends on which CRISPR enzyme, Cas enzyme you're using. But in this simplified model that I showed here, it does create a double-stranded break in the DNA.
How does that spread?
Yeah, so what would actually happen is you would most likely, if you wanted to create a transgenic plant or a plant with a specific edit, you would actually mutate a specific small group of cells called a callus or protoplast, and you let those grow up into a full plant. So basically, it cuts one strand of this DNA, or it cuts the two strands of DNA, and when that cell replicates, its daughter cells then also have that change. So that's how it gets transmitted to the whole plant. Yeah, of course. Yes.
We talked about how so cells don't like to have DNA fragments, and so they fill it in with kind of that green mutation layer.
Mhm.
Are you able to decide what fills in that particular spot, or is it just literally kind of random chance of whatever the cell decides to do?
Yeah. So again, that is very dependent on the type of Cas enzyme you're using and the system of CRISPR. Because in this very simplified system, you don't get to choose. You just cut it and hope something happens. And in many cases, actually, it repairs perfectly and you get no edits. But there are new versions of Cas that actually allow you to input new, like specific nucleotides. I forget exactly how many you can do, but it's called prime editing. And it basically allows you to transcribe in the system exactly what nucleotides you want up to a certain amount. Yes.
Seems like the normal shuttling protein is like pretty small that moves through. So are there like space constraints on how big of a thing we can use that to like move?
Or like, are there size constraints on the size of like things we can get into the cell with the shuttling? Or yes, and that's more, to my knowledge, more dependent on the size of the pores in the plant cells themselves as opposed to the peptide's ability to get things in. And that's another kind of offshoot of the research being done in my lab, is trying to figure out what role the cell wall plays and which specific parts of the cell wall are most restrictive to getting proteins into the cells.
Guide RNAs to increase drought.
So my lab specifically isn't actually working on mutating those genes specifically. We're more so working on the technology for how to get efficient editing. And so we would likely pair up with a different lab if we wanted to test this method out for them to use. But we also have a lot of like industry collaborators that are interested in using it for whatever genes they're targeting actually.
By immersing some concentrated solution of like the cutting of like the vehicle that you're transporting, that you're using to transport this cutting technology, or like how is it done at like a large scale?
Yeah, so this is still pretty new to the field, and so to my knowledge, no industries have really gotten to the point where they're scaling it up. We're still really in early stages of developing it as a technology for plants at all. But yeah, I imagine that, I mean, how we do it in lab is we inject the plant leaves directly with the solution. So I imagine potentially in a more industrial situation, they might develop many of these small callus of plants and then maybe drip the peptide on top of them with whatever protective thing. People have also talked about like in field, as far as like instead of using pesticides, you can increase the plant's ability to upregulate defense mechanisms by spraying this as you would a pesticide on the field on the plant leaves. Yeah.
Cell penetrating peptide to degree.
Many different ways, but for this, we specifically use a plasmid that just kind of had the Cree protein on it and translationally fused the peptides onto one of the terminals of it. Yes.
See how you develop the technology to get it on a cell wall. It's pretty sneaky, to use your word. But then you also get it into the nucleus of the cell.
Mm-hmm.
That's probably easier, but maybe different magnets.
Yes, so it is a little bit easier because oftentimes when you order this Cas9 protein, actually, they come with tags called nuclear localization signals. Specifically, so once it gets into the cell sees that tag and shuttles it directly into the nucleus where it can then do its cutting.
A little bit broad.
Okay.
Consumers generally still seem suspicious of GMO foods. Do you think we should overcome this bias?
I personally do, yes, and that's part of why I like to do these public outreach events, is because I just kind of want to get people more informed about the research and like what's actually going on in the lab. But yeah, I think overcoming that bias is a really big hurdle for research like this.
When it comes to the CRISPR modifications, are drought-resistant plants still theoretical, or have some been designed and created?
Yeah, I could not tell you specifically like a gene that they're working on currently, but I've seen many papers that are more on the other side of this, where they're looking for those traits specifically and then knocking them out or upregulating them to see if they do increase. And so, yes, that's not something crazy far off. People are working on this currently, and it's very feasible.
And follow-up would be, do you have an idea of the lengths that you can take, especially talking a drop of water and the plant's good, or I guess what's the possibility?
Yeah, I think that would be, best case scenario is that we could just have a very little amount of liquid, drop it on the plant, and it kind of shuttles itself through and does what it needs to. But the technology is still a little bit far off. We're still struggling, not struggling, but we still see quite low efficiencies. Like, as you can see here, it looks like only about 20%. Delivery efficiency. And so I think there's a lot more basic research that needs to be done to develop the system further before we can really use it for application. But I think it has some really big potential, especially considering that it's using like the plant itself specifically to do the engineering, essentially. Yes.
Seem to be very knowledgeable about what's going on. And really, by having the privilege of being a senior and totally ignorant in biology, but how can I tell you something? Apparently today there was a Persian New Year celebration somewhere around here. In Farsi language, Kas means someone.
is usually referred to a relative. And then the opposite of it is BCAS. It means you have nobody, somebody that has nobody. And then there's an expression that wants to cover everybody, and that expression is castle BCAS. Sorry about my ignorance being, diverted into a stupid joke.
Yeah, no, that's really interesting, actually. I am honestly not sure, unfortunately, where the name for CAS comes from, but maybe there's some relation.
I wrote it just in case. Sometimes it comes you something, this sort of information.
Interesting. Thank you very much.
Thank our speaker again.
For that wonderful presentation. Our second speaker today is going to be Kibby. She grew up just north of Madison, Wisconsin, and moved from there to Swarthmore College, Pennsylvania, where she received BAs in both biochemistry and Spanish. For graduation, she went to Boulder, Colorado, to get a PhD in chemistry regarding biochemistry, under the mentorship of Dr. Aaron Whiteley, who was the first person to love phage. Emily then moved to Berkeley in 2025 to pursue postdoctoral research with Dr. Kim Seed in the Department of Plant and Microbial Biology at UC Berkeley. Outside of the lab, she enjoys hiking, reading, climbing. So please give up that one.
Okay, just give me 1 second. You probably all don't want to read my notes to myself about how to do a good job on this talk. Okay, almost there.
Make it more personable.
Well, I can share the slides with you later if you want to read my script, and then you can tell me how good of a job I did at sticking to it. Yeah. Okay, there's one more. All right. Hello, everyone. Thank you so much, first of all, to the organizers for setting this up and running the show. I don't know how much the public realizes, but at this stage of my career, this is a very unique opportunity to get to talk to non-scientists about the things that I care about. A lot of research for your PhD and your postdoc is talking to like the same 10 wonderful people, but like the same people all the time about what you're thinking about. So getting to share with a different group is a really wonderful treat. And so I'll just kind of get right into it. So what really motivates me and what I think is really fascinating about science is this idea of something called a biological conflict system. So what do I mean by that? We can, for the purposes of this talk, we will broadly define biological conflict as antagonistic interactions between living things. I'm going to put living in quotes here because in the title of my talk, I spoil it that I'm going to talk about viruses, which are not technically alive. We can get into that debate if people care about it, but for the purposes of this talk. And so kind of, what I think about and the questions I'm asking day to day can broadly be binned into two categories, right? Which is, how do these living things threaten each other? And also, how do they protect themselves from threats? And just to kind of sort of zoom in on the scope of today's talk, I'm not so much talking about the interaction between like a lion and a gazelle, which certainly have an antagonistic interaction. I would more thinking about what we might often refer to as invisible threats. So does anybody have like a favorite example of an invisible threat, perhaps from 6 years ago? COVID, yeah, exactly. Infections. So these like microbial, what we often think of as microbial infections, so things that can make us sick that are caused by teeny tiny organisms that we can't see. But what you may not know is that the same things that can make us sick, so especially bacterial infections, so hopefully nobody's ever had food poisoning or like E. coli infection or something like that, those same bacteria that can make us sick also can get sick. They can be infected by viruses. And so this war that I'm going to be talking about is the war between bacteria and the viruses that infect them. And so this is a nice cartoon of like the major players in our game today that I'm going to talk about. So on one side we have the bacteria, and then on the other we have these viruses that infect bacteria called bacteriophages. You can't see it. This is not to scale, just to be clear. The phages are a lot smaller than the bacteria. But that's what we're talking about today. And so why does my title talk about the 3 billion year war? So I'll just give you a quick, brief history of the evolution of life on planet Earth. The planet forms about 4.5 billion years ago. About 1 billion years after that is when bacteria first appear. So these are single-celled organisms that we often think of as less complex than things like eukaryotes. I personally take issue with that way of viewing them, but we can think about them like that for now. And it's pretty safe to assume that around the same time that bacteria appeared, viruses that could infect bacteria also appeared, because if I've learned anything through grad school and my postdoc, it's that where there is life, something is trying to parasitize it. And that's probably also true for bacteria. And then for the next 3.5 billion years, a bunch of other stuff happened that we don't care about today. We'll just gloss over that. Okay, so I've been throwing out this word phage. Has anybody heard of a bacteriophage before? A couple people, yeah, okay, well, I talk to you about this all the time. This is my fiance. Right, but so fundamentally, you'll hear me call it phage. The technical full word is bacteriophage, but it doesn't roll off the tongue as well. Is a virus that infects bacteria. And so phages, just like any other virus, right, are effectively a nanoparticle that deliver genetic material into a bacterium and basically hijacked all of the machinery inside that bacteria and turn it into a factory that will make more viruses. And so I have just a couple images of what they look like here. So for context, this scale bar here is about 100 nanometers. So phage is about 100 nanometer. That's about 100 times smaller than the average bacteria. And it's so small, in fact, that we can't use normal microscopy, like, whatever microscope you used in like your high school science lab or last time you looked through a microscope, you wouldn't be able to see a phage with it. You have to use highly specialized equipment that actually uses even smaller forms of energy called electrons to visualize how tiny these are. But aren't they beautiful though? So we've got at the top here, just to give you an idea of the structure, we've got this, what we call the capsid. It's the head of the phage, and that's where all the DNA or RNA, whatever the genome is, lives inside there. And then it's got this tail, a lot of them, they don't all have tails, but this one does, that is used to inject that genome into the bacterium. And then we've got another image here just to get a little bit more scale. Just gonna keep tripping on it. Where you've got the bacteria. This is the membrane of bacteria, so the outside of the bacteria is here. You can see there's a lot more bug that we're not showing, and then we've got several phages adsorbed to the outside. You can see these like little green lines are where the DNA is actually being injected into the bacteria. So these are just some of my favorite pictures of phages, but now you know what we're thinking about, what we're looking at. And then kind of the other, I think, important scale for thinking about this conflict is that phages are actually the most abundant biological entity on the planet. There's an estimated 10 to the 31 phages on planet Earth, which is 10 million trillion trillion. If that number means anything to you, it doesn't really mean anything to me. I can't conceive of a number that big, but I can tell you that it's more than any other organism on the planet. That's more bacteria than there are in the world, and it's more stars than there are in the sky. So just for some context about how abundant these little microbes are. That's a great question. We were just talking about this the other night. They have a little bit, I think they've just had a little bit of a reputation problem, but we're working on it. And so I'm glad you're here, and now you can spread the word. Because I'm hoping to convince you today that phages are really cool and also really important. But we've known about them for decades, but they got a little overtaken by antibiotics, because antibiotics also kill bacteria a lot. And so the Western world, really everything outside of like former Soviet Union has largely preferred and studied antibiotics for a long time, and maybe not given phages their due, but they're having a renaissance, I would say, right now. So it's an exciting time to be in phage research. But okay, so why, besides the fact that they look really cool and are super abundant, how does this 3 billion year conflict that bacteria and phages have been undergoing actually shape our world? And so kind of broadly, the thesis of this, right, is that, well, phages are super abundant. Bacteria are also super abundant. I don't know how many of you regularly attend these talks, but a couple months ago, there was someone talking about how microbes and bacteria are really found everywhere in the world, any surface you can think of, and living pretty much anywhere you can think of, and doing all kinds of fascinating things in our gut, in the soil, in the ocean. And because bacteria can have these really impactful effects on biology, from the carbon cycle to gut health, and phages control how bacteria can grow and what they can do. And so really this dynamic regulates a lot of important biological processes. And so for the next part of the talk, I just want to give you a few more concrete examples of ways in which I think this conflict is cool or maybe particularly interesting. And then at the end, I'll give a little bit of a taste of what I am specifically working on and some of the tools that I think about for solving that. And again, want to highlight interactive discussion. Please jump in with questions at any time, especially if anything's confusing. I want to bring everybody along for this ride, so please ask me questions. Okay. So our first example of how this combat matters is in thinking about the marine carbon cycle. So we love trees, right? Berkeley's a beautiful place, got a lot of trees. They make oxygen for us. But actually 20% of the oxygen in Earth's atmosphere is produced by bacteria, not the plants that we know and love. And most of these live in the oceans. What we also know about this microbial warfare in the oceans is that something like 20 to 40% of all the bacteria in the oceans are killed every single day by phage, which is a huge amount of turnover in a living organism. And when these bacteria die, the particle, they effectively burst open. And so that debris becomes part of marine snow, which you may or may not have heard of, which is the detritus of living things that have died in the ocean, basically. And it's an important food source for other bacteria and lots of organisms in the ocean. And it also serves as kind of carbon pump or carbon sink, because all of the carbon used to build those bacteria is sequestered and falls to the bottom of the ocean, where it stays there for decades instead of being in our atmosphere. And so this interaction between the phages and the bacteria really regulates a lot of this turnover. Okay, example one. A second example is a little bit different in thinking about how bacteria influence, how phages influence bacterial pathogens. And this is actually a little bit less of a antagonistic example and more of a cooperative one, where the phage and the bacteria are working a little bit more together. And this is talking about my favorite pathogen currently, cholera, which is not really a big problem in the US, fortunately, but it does still affect the lives of millions of people in the global south and kills thousands of people every year. So still very much a present threat. And how it does that is because cholera produces a toxin that, when ingested by people, basically makes them have really terrible diarrhea. And they get super dehydrated, and that's how they can become very sick. But what's fascinating about this pathogen is that the gene, the piece of DNA that says make a toxin to make people sick, is not actually from the bacterial genome. It's actually encoded by a lazy phage that has gotten into the bacteria and just is kind of hanging out there. And in the meantime, the pathogen, while it has this phage, is making this toxin. So if you have, and we probably have some in the lab, I'm looking at one of my lab mixes here. If you have a bacterium that made people sick, right, that can make this toxin, and you remove the phage from it, now that bug is not going to give someone diarrhea if they were to ingest it. They might have some other problems, because generally you don't want to be drinking like a ton of bacteria if you can avoid it, but they're not going to have cholera to nearly the same degree. Okay. That's a great question. That's the question. And I think it's a gray area, right? 3.5 billion years of war. That's a lot of space to explore in what kind of tools you want to use to fight each other, and maybe you want to cooperate sometimes. And so there's unfortunately not the right answer, which is why we have to keep studying them so we can figure out which ones are good and which ones are bad. Well, there you go then. Yeah. It's a great question though. Okay, So that's another example. A third example I want to bring to your attention is sort of the industrial uses of phage. So humans are not really bystanders in this conflict. Not only do we live in the world that's impacted by it, but we do also use it to our own advantage. And so a couple of examples of where we use phages already is that we can use them as pesticides to protect crops from specific bacterial pathogens. And they can also be used as sort of decontaminants in food processing facilities to target things like listeria or salmonella, things that can cause really unfortunate outbreaks. And the reason that phages are so useful for a process like this is because they're highly specific to their host, generally. So if you use phage as a pesticide, you can target just the bacterium that's making your plant sick, while you will not be impacting, either the neutral or beneficial bacteria that might also be on your crops. And they can also be very efficient, right? When they are successfully able to infect a host, one phage can lead to hundreds of viral particles that will then go on and do more infections. So when this infection is successful, it can be an incredibly efficient way to clear dangerous bacteria. On the flip side of that, those same features that make phage so good as a pesticide or a decontaminant also make them a nightmare for industrial processes that rely on bacteria. So does anyone have favorite examples of something that's made in a bioreactor or something we might use that relies on bacterial cultures? No, okay, fun fact. So lots of fermented products, so things like cheese and yogurt rely on being able to culture bacteria in a controlled manner to ferment these products. Also things like insulin. Right, some of you may know people who have diabetes and need to use insulin or monitor insulin, and it's produced by bacteria who we've, using tools like CRISPR that we heard about earlier, we've told these bacteria make a bunch of insulin, and they're really good at it, and you don't have to pay them overtime, and it's great, and it's a great way to produce this product, which is why insulin should be cheap. Anyway, that's a different topic. But you can imagine if you have a large, vat growing a bunch of bacteria that are all the same, that are all doing this one thing that you've grown them to do, and you have a phage come in, it can really easily wipe out and kind of decimate your production pipeline. All right, that's my industrial example. And then my final example of one of the ways we can use or better understand this conflict is in treating bacterial infections. So hopefully none of you have had to deal with antibiotic-resistant infections, but it is an increasing problem in the US and throughout the world that people are getting sick with bacteria that are not responsive to normal antibiotic treatments. And that's because they've acquired different genes, different mutations that mean that antibiotics don't kill them the way that they're supposed to. And in some cases, phages have been used to successfully treat people who are infected with these resistant bacteria. And so right now, it's a really kind of burgeoning area of research is trying to understand how to effectively use phages as therapeutics to treat some of these resistant infections. And the schematic here is just kind of highlighting, so really the most pertinent bar is at the top here. And this is from a study done a couple of years ago, but it's estimated that in 2020, just over 1 million people were killed by bacterial infections that were resistant to antibiotics. And without some major changes going forward in the way we treat infections, that number is predicted to be as high as 10 million by 2050. So I don't mean to alarm anyone. I don't mean to be alarmist, but it's definitely something to keep an eye on, something to think about, and that's why we're studying phages. And so that kind of brings me to what my area of study is and what I've been thinking about a lot for the past, when did I start? 6 years. And that is thinking about how bacteria defend themselves. Because I have so far presented this conflict as largely one-sided, right? The phage come in, they wipe out the bacterial population, and the viruses are winners in a lot of the scenarios. But that's not really always the case, right? As I said before, the bacteria don't take this lying down, and again, they've had 3 billion years to evolve defenses to this infection. And hopefully by now I've convinced you that the outcome of this combat, of whether the bacteria lives or whether the phage lives, can be really impactful in a lot of different situations. And so what I try to study now and what a lot of people are working on now is trying to understand what are the features that determine who lives and who dies, right? What are the traits about a bacteria that decide whether it's going to be susceptible to infection, and how can we use that information to better design phages as therapeutics or as tools. And so in addition to kind of that impact or that overall goal for a lot of this work, I do just want to highlight a couple of the more fun pieces of research that I think have come out in the past decade or so as we've been identifying and trying to understand more and more ways about the things that make this conflict really tick. And so a couple of them I've listed here. This is just kind of a brief little schematic of what the phage life cycle is. I'm happy to go into it in more detail if people are interested in. I thought it was a nice visual from our friends at the Innovative Genome Institute here at Cal. But so some of the things that we've learned in the past decade studying phage defense. So far, we've identified over 100 different genes in bacteria of all shapes and sizes that can confer resistance to phage. So this, paints a picture of an incredibly diverse and incredibly widespread immune system that bacteria have developed. We've also found that some of these genes are actually ancestral to parts of the human genome. So some of the proteins and genes that we use in our own immune system that protect us from viruses or bacterial infection actually originated in bacteria, and we adapted them at some point in our evolution for our own purposes, which I think is very cool. And then one of the last things is that, in and of their own right, because bacteria have had 3 billion years, not to harp on it too much, but that's a lot of evolutionary space to explore, to innovate in this fight. They've done some really incredible biochemistry. And so we had a wonderful talk about CRISPR just now, so I don't need to explain it again or why it's important or cool. But in the wild, CRISPR is a way that bacteria protect themselves from phages. It originated in bacteria. People identified it originally because they were trying to help their cheese fermentation stay alive better, and they identified these CRISPR genes as ways that made bacteria more resistant to phage infection. It's been turned into this incredible tool. Okay, and so lastly, does anybody have any questions? I don't know how I'm doing on time. I think I'm okay, but okay. So those are the questions I'm thinking about. That's what I've been working on, and what a lot of people are working on right now is trying to understand this conflict and some of the features that are really determine which way it goes. And so I'm just showing some of the tools that we use for that. These are cartoons. Obviously, we do a lot of gene editing. Some of it is CRISPR-based. We have some other tools working primarily in cholera that we can use to edit the bacterial genome or the phage genome and more directly test what features might be involved. This, of course, also relies on a lot of sequencing data. So really being able to get a whole picture of what is the genetic material of the different organisms we're looking at. And then all of this really leads to my favorite experiment, which are phage assays, which are my favorite thing to do. And so what this is a really direct way we can quantify how well a phage is able to replicate or how well a bacteria can replicate when it's challenged with phage. And so what you're looking at here is just some pictures of plates, like the ones that are laid out here. And what you can see is this dark area is these are alive, growing bacteria that are happy and healthy. And all these clear spots are actually dead bacteria, where a phage has successfully infected and replicated. And what you can see is these are 3 different conditions, right, testing with different bacterial mutants with the same phage. And we can see that they all kind of have different experiences in this conflict with phage, right? So this one in the middle is clearly having the worst time. The phages are doing great. They're replicating super well. Whereas this one over here is able to protect itself a lot better. And we can study how that protection happens. And so that's all I've got for you today. A quick thank you to everyone in the Seed Lab and also my old lab at CU Boulder, which is where I got into phage in grad school, the folks at the library and Popping the Science Bubble for putting together this incredible thing. And thank you all for your attention and taking the time to be here. It's really appreciated. So any more questions? Yeah. Oh, so many. I think you had yours up first, and then we'll go over there.
So do all bacteria have phage?
I'm going to say yes. We haven't identified phages for all bacteria, but I think that's probably that we haven't looked hard enough. I think probably all bacteria have a virus that infects them.
So categories of phage depending on
Yep.
the bacteria?
Yes, yeah, there's all different kinds. Phages are, I would say, equally as diverse, if not even more diverse than the bacteria that they infect.
So, picture that you showed,
Mhm.
was like super small.
Yeah.
On there, is that about the smallest that we can see?
No, we can see smaller things. We can actually see, I don't have any good examples for you, but I like that picture of phage, so we'll go back to it. We can actually look at things as small, like DNA. The DNA inside of that we've solved the structure with that using other techniques. We can visualize the structures of different proteins, but this is certainly on the smaller scale, but not the smallest, I would say. Yeah, absolutely. Amazing question. Incredible question. Yes, there are parasites of phages. There are even smaller phages. We often call them satellites. This is actually also an active area of study in the seed lab at Berkeley right now. They are elements that live in the bacterial genome. And then when the bacteria gets infected, this satellite element instead hijacks the phage replication to package and spread itself. Yes, that was also my reaction when I first learned about it. It's really incredible. Yeah, I don't know how far down this parasitism goes, but certainly there are parasites of phages. Yeah, it's very cool stuff. Thank you for that question. Glad I got to talk about satellites. I think there was a question over here as well. Uh-huh. Yeah. Mm-hmm. I think it really highlights how quickly happy bacteria can grow. Is because I don't think that the overall population of bacteria in the oceans is declining, at least not due to phage infection. And they are, able to recover that amount of their population every day. Exactly. It's I don't know how finely tuned the balance is, but there's definitely a balance going on. All the marine bacteria, yeah. There are phages that infect cyanobacteria. There are lots of things that are not cyanobacteria that also live in the ocean, and they are also getting infected. So it's I would say probably across the board. Yeah, great question. Yeah.
Like, could there be eventually phage resistance?
Exactly. Yeah, this is a wonderful question. Also, thank you for asking it because now I get to talk about it. Something I think about a lot is, yes, there is definitely the thing. And I think that's one of the reasons why adoption of phages in the clinic has been so slow. But I think, and if you talk to certain phage biologists, some people think they're really not a good idea to use as therapies. And another, everybody puts it in their grants because it's exciting and people are interested in it. And I think whether or not we should is probably the wrong question because the cat is out of the bag, right? If I'm really sick with an antibiotic-resistant infection, I don't want to tell a clinician what they can and can't use to save my life. And so I think the question is not should we be using them? The question is, what do we need to know to not put ourselves in the same place that we are with antibiotics right now? And I think we've got an opportunity because we have so many tools and we know so much more. We're in a better position to prevent that than we were when we first discovered penicillin, right? So we kind of get a do-over, is my hope, is that, we're starting at an early enough square that we can not make the same mistakes and we've got time to learn what it is we need to know to use these things safely and effectively. Oh, sorry, you're next. I'm not forgetting. Ooh, that's an interesting question. I like that idea. I don't know that anybody's done it. So you can definitely, there's been an amount of work aimed at engineering phages to change which bacteria they can infect and understand what determines that kind of specificity. I don't know that anybody has done it to a virus. I think part of the problem that it might be challenging is that the reason phages are so harmful to their hosts is because the host has machinery that the phage needs. But all the host machinery that the phage is like taking over and co-opting is required for the host to continue being alive. And were the virus to infect another virus, there's not that same pull. It would just be like, well, we're both stuck here together, not able to do anything. But that's a really interesting idea. And because they can be so specific, there's kind of an appeal. I didn't talk about this, but people have used phages as a way to kind of as a screening strategy. If you get like a stool sample from somebody or some kind of other environmental sample and you want to quickly screen what bacteria are here, some people will use like a panel of different phages as kind of indicators. And you could imagine maybe trying to develop them into something like that for viruses. That would be very cool.
Also, this is a fascinating talk, so thank you for.
Thank you for being here.
And yeah, I also had a question about how phages are killed. And you mentioned the satellites and parasites and phages, but I'm wondering for instance, the industrial life that you talked about, phage like bacteria colonies, what they use to kill the phages, or do they use different things industrially?
I love the idea of using satellites industrially to kill phages. That's not how they do it, but it would be really cool. No, I mean, so a lot of time it depends on what kind of phage is contaminated with. I mean, bleach kills all, right? But also sometimes things will like UV irradiation because they are, fundamentally just packaged DNA or other nucleic acids. UV irradiation to damage that genetic material so that even if they were to successfully infect, there's no way they could replicate. It's all just nonsense in there. Is another strategy. And then there are other, sort of chemical agents that can be used to pop open the capsids so that they're just free-floating materials, basically. Yeah. Is there a question over there?
How long or how they do this without the host?
That's a really interesting question that I don't have a good answer to, and I think it probably varies quite a lot depending on the phage. Yeah, the short answer is I don't know. The second answer, the long answer is it probably depends a lot on like the physical properties of this particle and the environment that it's in. Is it being washed around in the waves? Is it just sitting on a rock? Is it getting UV irradiated by the sun? I think all these things probably impact how long the phages can live. I know we've got some like pretty old stocks in the freezer, but so if you keep them really, really cold and store them in the right media, they can be viable for a long time. But just like out in the environment, I suspect it's not nearly as long.
Can you talk a little bit, so like what makes a phage so specific to a particular bacteria? Because I imagine like there's a lot of characteristics that are similar across bacteria, like why they don't then cross over a whole lot.
Yeah, that's a wonderful question, and one that honestly I think we as a scientific community don't have a complete answer to you. So I'll give you the best picture I can with the data that we have. And there are a couple of factors. One of them is, so you're absolutely right. There are a lot of shared features between bacteria, but there is also, sort of the physical features on what is on the surface of bacteria can be quite variable. And so a lot of phages are very specific to recognizing like one protein or one weird like sugar structure that their host makes. And that's what they bind to, and that's how they can attain such specificity. The second part of it is I do, I'm a little bit biased because I study bacterial defense systems, but I do think bacterial defense systems play a part in that specificity, in that if you have two very closely related bacteria, but one of them has a defense system and the other doesn't, only one of them is going to die, right? So I think those are some of the factors. So these like outer proteins that are kind of on the exterior of the bacteria are a large part of the specificity. And then there are also sort of internal components that determine how well the phage can replicate. But it's a great question, and one like a lot of people are thinking about. There's some really interesting work being done trying to understand that. I'll go over here first. Yeah.
I was amazed to learn that in cholera, the toxin is coated
find things.
Mm-hmm.
Maybe this is all news to everyone.
No,
Is
probably.
that a recent
Oh, well, it's not a recent finding, but I don't expect the general public knows it.
Or is it something that you might think that there are another bacterial pathogen, so it's actually a phage that's causing the disease?
Yeah, that's a really wonderful question. So I'm glad you asked it. So I think the example of the cholera specifically encoding a toxin is a little bit unique. There are other examples of things that are actually these satellites that I talked about, these parasites of phages, also encode toxic molecules in Staph aureus. But by and large, I would say most of the things we would call virulence factors, which are the things that the bug makes that make people sick, are generally components of the bacterial genome. However, we do also know for some of these, what I called lazy phages that can kind of hang out inside of bacteria for a while before replicating, they do carry all kinds of interesting, what we call cargo genes with them. So things like the cholera toxin is kind of a more exciting example. Some of them actually carry defense systems because if you're a lazy phage, maybe you don't want your host to get infected by another phage before you've had a chance to finish replicating. So there is definitely interesting cargo. I wouldn't say that it's the most common way for bacteria to make people sick is by a phage thing, but like I said, it's a little bit more exceptional, but right, like, you can't make any hard and fast rules about bacteria, I feel like, because they're so diverse, they're so abundant. There's so much that could happen, could be happening, but it's a great question.
Yes, someone asked, it's surprising in a good way that certain viruses are approved for use in food. Are viruses subject to the same environmental constraints like moisture and temperature as the bacteria they parasitize?
That's an interesting question, and it kind of goes back to something I can't answer completely about this stability. And I think probably the answer is some of them yes, some of them no. Some of these pesticides, I imagine a lot of the work in optimizing some of their use as decontaminants and pesticides has partially been, what do you need to mix the phage with in the spray bottle so that it will remain stable and effective for the longest amount of time. And I'm not sure, but it's a really interesting question. There are probably some physicists at Berkeley, honestly, thinking about it, and some engineers. Yeah.
Do we have a battle of phage and bacteria going on in our bodies right now?
Absolutely, everywhere, on your skin, in your gut, everywhere. I think definitely some people are thinking about how to use phages as not probiotics, but as ways to sort of modulate your gut microbiome as we kind of learn more and more about how the health of the bacteria in our gut can impact other ways of our existence, and using phages to kind of tinker with that. I think we're probably, I don't know that we're there yet, but certainly people are working on it.
So who's the overseer or who's the referee in this battle to make sure that it will kind of stay stable and one side doesn't overpower the other, and then we're just throwing all out?
That's a fascinating question. Can I answer it? Yeah, I mean, I don't think there is an overseer, really. I mean, I think the fact that it's been happening for so long is what has kind of allowed us to reach this sort of homeostasis, if you will, or this type of balance. But that's not to say that certainly if you eat something weird or like introduce other bacteria into your gut that maybe weren't there before, that homeostasis can't be like thrown out of whack.
Right. Killing each other off at the same rate?
I mean, we have to assume it's approximately the same rate. I don't think there's any evidence that we're like all the bacteria in the world are dying, and that we're like losing bacteria overall. So there is certainly like a homeostatic interaction going on. And what you can think about it is true for a lot of, either predator-prey or virus-host relationships. If you want to think about it, it's disadvantageous for a virus to completely wipe out every member of its host, right? Because then the virus will never be able to make more copies of itself. Yeah, exactly. So there is kind of a balance there, right? Just like it would be disadvantageous for a pride of lions to eat every single gazelle in their hunting grounds, then they will never be able to hunt a gazelle again, right? So maybe that's not the best analogy. I don't study animals, but there's certainly a balance. Yeah.
Why do they both exist in the first place? Like, why even have this battle going on?
It's an amazing question, and also one of the reasons that I got into science and why I love this so much. Because if you zoom back far enough in our evolutionary history, I think we're all here because like some pieces of DNA decided they wanted to make more copies of themselves, and then from that evolution over billions of years, we have this eternal war with bacteria and viruses, and they're doing their thing. But also we've evolved so many other things, and kind of fundamentally, it's all just because at some point genetic material started wanting to make more copies of itself. And here we are 4 billion years later, thinking about it, talking about it. It's a wonderful question. I don't have the right answer, but yeah, it's sort of a philosophical debate, but it's a good one to think about. And one of the things that keeps me coming back to research, keeps me coming back to science is, the miracle of life, however you think about it. I think the fact that that's where we started and that's where we are today is kind of miraculous in and of itself, even if it was messy and confusing along the way to get here. Yeah, thank you guys.
Mm-hmm. Last question. If these phages aren't alive, how do they know where to migrate? Like, is it just like an energy landscape or like convection?
Oh, sure. Yeah. So I'm going to say it's probably most of the time like, kind of just like physical diffusion. They just go out. There is an amount of, so for a lot of phages, when they infect and they replicate and they make copies of themselves, they basically fill the bacterium to bursting with copies of phage. And then there is a sort of violent event where the bacteria pops open and the phages are kind of ejected out because of like the force of how packaged they were. And so that, will propel them some amount of distance depending on where they are. And then they probably hope that they interact with another bacteria along the way, or they stick to somebody's shoe and get carried along in much the same way that many microorganisms move about. But I don't think there's not much evidence for bacteria like, sensing some signal from feet away because they're very small and being able to specifically move towards it. There might be interesting ways that like the morphology of like their physical shape allows them to move in different environments, but I don't know the specifics of that. It's a really interesting question. Okay.
Speaker again.
Thank you. Thank you.
For our closing, I will put up the slide once again. But thank you so much for coming out to our Popping Science Bubble seminar from March, and thank you to those of you who are joining us online. If you have enjoyed hearing these talks and are not currently set up to hear about our future talks, we have this very handy dandy QR code that you can scan over here to sign up for our email list. Additionally, we also post on our Popping Science Bubble Facebook page if you are active on Facebook. And then, as we mentioned before, thank you, Molly, that there is also our YouTube page that this seminar will appear on, and future seminars as well. So our next seminar is going to be April 21st, so just about a month's time. And we're going to be talking about fermentation ecology. I don't know what that means, but I'm excited to hear about it. So until then, thank you again to our wonderful speakers for talking with us. And let's give them one last round of applause. We hope to see you in a month's time. Thank you so much for joining.
Thank you so much. Thank you. Thanks again, organizers.